All right, I would like to welcome everybody to the fourth Monday of this year. Denny Anderson, Dr. Anderson will start. He's going to be the first one. Denny Anderson went to Indiana University Medical Science program with a major in comparative physiology. This is where he got his PhD. He did a postdoc at the University of Wisconsin School of Veterinary Medicine. And before coming to us, to North Carolina Wesleyan, he was an assistant professor at the Department of Kinesiology and Health Sciences at Hendrix College in Arkansas. The topic of this talk today is breathing during exercise, what we do and why we do it.
Yeah, I was telling my colleagues that I wasn't really nervous about this until everybody told me how much they were looking forward to it. But anyway, so what you're going to get today is somewhat sort of a research journey, okay, where I've been. And I'm going to start with my postdoc research, where we looked at something called serotonin-dependent modulation. Okay, so I need to make a distinction between modulation and plasticity. So with modulation, you have a cause, and as long as you have the cause, you have an effect. Okay? You take away the cause, you no longer have the effect. You reintroduce the cause, and you have the effect. Okay? So in this case, the cause is going to be serotonin. Plasticity, an example of that for you psychology people would be long-term potentiation, where you have rapid stimulation of the neurons, the Schaefer collaterals, and the CA1 region of the hippocampus. And once you do that, okay, after that, you get a stronger firing, okay, of those neurons with subsequent stimulation, and that's the cellular analog of learning and memory. What we studied was serotonergic modulation of breathing during exercise. Okay? And so I'm going to start by asking a question. When you hold your breath and you want to let go, okay, there's a drive to breathe, and eventually it's like, does anybody know what that drive is? Can anybody guess? CO2. Okay, thank you. Okay, yes, that sounds like a college professor answer. It is indeed buildup of CO2. Okay, so your breathing is regulated to tightly regulate levels of arterial CO2, mainly because it's tied in with acid-base regulation of the blood. Okay? If you get oxygen really low, then it can drive breathing. But for us right here, what's driving breathing at rest is arterial CO2. Okay? So to really look at control of breathing, you have to look at arterial PCO2 specifically, CO2 pressure in the blood, in arterial blood, okay? Because it's sensed by your carotid bodies up here, and that's the blood going to the brain. So regulating blood gases and pressure is important. So those receptors are important. Okay, so when you start exercising, do you breathe more or breathe less? Thank you. Okay, that was an easy question. Okay, that was insulting your intelligence. Okay, so yeah, you breathe more. Does anybody know why you breathe more?
You need to take in more or something.
You need to take in more. That does help. Okay, and what is so the drive to breathe at rest is arterial CO2. Anybody want to guess what it is when you exercise?
Is it to keep your cells going, like your blood cells?
Okay, what is the signal? So are you saying it's feedback from your cells to the breathing centers? Or?
Like getting more oxygen to your body to keep the the way it's supposed to. I can't remember the intended.
Okay, well, that does happen, and it does help. We're asking, like, why does your breathing increase? What is the trigger? What is the cause? Anybody?
Because your body's working harder. So.
Okay. Okay. Well, what causes your heart rate to go up? Yeah. Okay.
Your whole body's moving more.
Your whole body.
You're exercising versus when you're at rest.
Okay, that is true. Okay. Why does that make your breathing increase?
Because you have to have more oxygen flow throughout the body versus when you're at rest.
So can you give me the signal to your respiratory muscles. Actually, you are on the right track. The fact of the matter is, you know, and some people would guess that, well, okay, it's buildup of CO2, you know, because you're producing more CO2, that's going to feed back and elevate your breathing. But what I'm going to tell you about, these are old slides, okay? My postdoc research, we studied something called short-term modulation of the exercise ventilatory response. So I will take you through. We start with a goat standing on the treadmill, okay? Any questions? Okay, good. Now, if the goat is alive, the goat is producing CO2. So what you have is inspired gas, and we actually gave the goats a hyperoxic mixture to make sure oxygen had nothing to do with stimulation of breathing, okay? So we measured, well, expired ventilation, and they are going to exhale some CO2, and their metabolic rate is proportional to their CO2 production, okay? So we also had an arterial cannula to take blood samples out of their carotid artery. So before any experiments, we had to do what's called a carotid loop surgery and bring the carotid artery up to the surface of their skin. So we could stick a big 18-gauge needle into it at the beginning of the day. They liked that a lot. But anyway, so that was one of the, anyway, we could take blood samples and run it through a blood gas analyzer and measure arterial PO2 and arterial PCO2. That was the most important thing for us. Now, a lot of people say CO2 when the goat exercises. Well, the goat, first of all, breathing increases, okay? If that were feedback from CO2, then you would expect to see an increase in CO2, but goats actually breathe enough, most mammals breathe enough to actually lower the amount of CO2 in their blood by about 2 millimeters of mercury. So there's no way it can be feedback from blood gases, okay? So what causes breathing to increase during exercise? Basically nobody knows, okay? Nobody has ever been able to experimentally have an animal exercise while blocking the exercise ventilatory response. The putative, that's a great word, it basically means that nobody knows what's going on, but the putative it's feed forward, okay? So the same motor program that tells your locomotor muscles to begin their activity also tells your respiratory muscles to begin their activity. And that's different for different people. Different people have different ventilatory drives during exercise, okay? So I used to breathe like a freight train, okay? Other people don't sound like they're breathing at all, okay? So anyway, and it's, by the way, it's best not to try and mess with that. Don't try to tell somebody, you should breathe less when you exercise, you know, but anyway. So that's the normal exercise ventilatory response. Now, if you add a demand, okay? So in this case, short-term modulation, so this is the normal exercise ventilatory response. Well, let's say we make it a little bit more challenging, okay? So this could be a, obstructive pulmonary disease, it could be weight gain, something like that. So what we did is we added some dead space to the end of the mask. What that's going to do, the goat's basically going to rebreathe the CO2 that it just exhaled. That's going to increase the amount of CO2 in the lung, and that's going to increase the amount in the blood, and that's going to stimulate resting breathing, resting ventilation in the goat. So what you see is an increase in resting ventilation, and resting arterial CO2 increases. Okay, so now you have the goat exercise. Well, the ventilatory response, when the goat was at rest, arterial CO2 decreased by about 2 millimeters of mercury. Okay, now when the goat exercises with the dead space, you still see that same decrease. But in order to do that, okay, the goat has to augment its exercise ventilatory response, okay? So it has to have a greater response to, breathing response to exercise to regulate its CO2 relative to the resting level, okay? So it's feed forward, okay? Breathing during exercise, it's feed forward with respect to whatever the resting level is, okay? If you change the resting level, the exercise ventilatory response will change accordingly to decrease by about the same amount relative to that new starting point. Okay, now, so as I mentioned, that can be what happens with an obstructive lung disease, something where it's harder to breathe, weight gain, restrictive lung diseases. So you need some kind of boost to the respiratory muscles in that case. Well, this is where we studied the role of spinal serotonin. Okay, I think we all know serotonin as, you know, the drug where you take Prozac. We actually did a study where we put goats on Prozac. But anyway, serotonin also has a motor function. So our model, okay, so short-term modulation, if we look at a graph, okay, well, this is resting arterial CO2, okay? So this is resting breathing, resting ventilation. So when the goat starts exercising, it increases the exercise ventilatory response, increases breathing sufficiently to lower the amount of CO2 in the blood, okay, relative to this resting level, okay? So with short-term modulation, well, you make it a little bit more difficult. You add the dead space that increases resting arterial CO2, okay? And when the goat exercises, well, we have an augmented exercise ventilatory response, okay? So a boost to breathing during exercise, then we see the same regulation relative to the resting level, even though the goat has to work harder to do it. Okay, so we have the goat kind of getting a stronger signal to the respiratory muscles in order to regulate CO2, same relative to the resting level. So impaired short-term modulation. Well, if we just use a mask, if we just have the mask, well, really don't see much difference. Because short-term modulation is that augmentation with the exercise ventilatory response. Augmentation. So if we add dead space, then what you see is less of an augmentation of the exercise ventilatory response. It doesn't have that extra boost to the breathing muscles, okay? And that's reflected in the inability to regulate CO2 when you have that extra demand. So the role of serotonin in all of this. Serotonin is involved, previous experiments. Well, if you look at the difference between these two yellow lines, the difference in the slopes, that's going to be 0, okay? If you look at the difference in the white slopes, okay, that's going to be a bigger number. So on the next graphs that I'm going to show you, okay, any number closer to 0 is going to be perfect short-term modulation. It's not going to be perfect, but a larger number is going to represent impairment of short-term modulation, okay? So we're going to basically be looking at the delta, 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 the change in the change in the change. So, okay, all this mess over here is just the difference in the slope. Dead space compared to mask only, okay? So methosergide is a non-selective serotonin receptor antagonist. It blocks all the serotonin receptors. So this was injected spinally, okay? So there were surgeries done to implant spinal catheters, little myodome reservoirs, and we would inject it. They did. These experiments were done before I got there. But when that happened, okay, well, there was an impairment of that extra boost during breathing during exercise. So in trying to get at, well, what receptor subtypes, where in the spinal cord is serotonin working? What are the important sites? Well, 5-HT2 receptors are excitatory serotonin receptors. They are present right there where the neurons synapse with the respiratory neurons going out to the breathing muscles, okay? Okay, so in terms of the model of how serotonin works, okay? Any questions? Yeah, not funny. Okay, so anyway, we got the brain stem and the spinal cord. So brain stem up at the stem of your brain, okay? And the spinal cord. So what we're looking at here is a cross-section of the spinal cord. And if you look at a cross-section of the spinal cord, it's highly organized, okay? You have specific tracks running in specific regions of the spinal cord. And in the middle of all that, so that's the white matter, because you have myelin sheaths, lipid sheaths around all the neurons going up and down to and from the brain. And in the middle of all that, you have the gray matter, which is where all the cell bodies are. So any connection between neurons coming down from the brain and going out to the body, or coming in from the body and going up to the brain, happen in the gray matter, okay? Okay, so in the front, you have the ventral horn. In the back, you have the dorsal horn. So in our model here, these are our brainstem respiratory centers. So you have the rhythm generator, the timing of the breaths, and then you have the pattern generator, the shape of each breath. Okay? So there's interaction between those, and anything you do to change breathing, you're changing your brain, you're affecting your brainstem respiratory center. So you have the dorsal respiratory group, the ventral respiratory group. Okay? So those neurons travel down the spinal cord, synapse in the gray matter, and send a signal to the respiratory muscles, the breathing muscles. So right here, also in the brainstem, okay, we have our raphe nuclei. These are the nuclei, the serotonin, where the serotonin neurons originate. And we all know about the psychotropic effect, but this has a motor and sensory modulatory effect. Okay, so right here in the ventral horn, well, okay, we have serotonergic projections. We have our motor signal to our respiratory muscles coming down, and there are 5-HT2 receptors. Those are excitatory receptors. So we saw that if we block those receptors, well, what modulation means is that it alters the excitability of these neurons. So what it does is it makes them more excitable. So this signal coming down from the brainstem respiratory centers has a greater effect. So when you need that extra boost, spinal serotonin is necessary. Okay? Okay, now, in the dorsal horn, well, these are our sensory afferents. So sensory input also modulates or modifies breathing. The problem is that the sensory input, so this would be proprioceptors. And proprioceptors are things like muscle stretch receptors, joint receptors, the kind of sensory reception that you're not really aware of. Most of you probably aren't aware of the fact that you are aware of where your limbs are right now, okay? But that's proprioception, okay? So anyway, if you alter that, if you start increasing limb movement, like you mentioned, well, that will feed back, but it'll actually have an inhibitory effect. So generally, when you're exercising, you could do without trying to inhibit your respiratory center. So in that case, we have other serotonin receptors, such as 5-HT1 receptors, that are inhibitory. So basically, you are inhibiting the inhibition. And we wondered about those. Do these have a role? Okay? So we did the same experiments, same set of experiments, only injecting a 5-HT1 antagonist to block these receptors. And these were most of the results of my postdoc. Okay? This is physiological data, folks. It's all over the place, but there's nothing there. Okay? No effect, no apparent role of 5-HT1 receptors in that short-term modulation. Okay, well, with that, we went to another hypothesis. So I wrote an NRSA grant and proposed with this model. We know that serotonergic projections are necessary for short-term modulation, for that extra boost. Now, in the raphe nuclei, they have serotonin receptors. They're autoreceptors, so 5-HT1 autoreceptors. So if we stimulate those, will it block the serotonergic transmission necessary for that short-term modulation? Okay? And we did the experiments, and we gave it systemically. Okay? So the goats kind of got a little freaky after we gave the drug, but we were able to do the experiments, and lo and behold, wow, okay, this is mask only, dead space pre-drug. Okay? Post-drug, mask only, they were no longer able to regulate CO2. So for one study in my postdoc, the results were actually consistent with the hypothesis, and I got a positive result. Okay? I was very happy about that. Okay, so, and here you can see this, the white with the squares, okay? A slightly flatter slope, okay, than the yellow with the squares, okay? So, decrease in the augmentation of the exercise ventilatory response reflected by an inability to regulate CO2, okay? Okay, now, I did other experiments with undergrads when I was teaching at Center College in Kentucky, and I got new equipment, and I was very excited about that. I will tell you about it in a second, but I was bringing this physics professor I was at Centenary College. I had been at Center, and a physics professor that I'd known at Center was coming to visit. So I was going to drag him down and show him our new equipment. And the Centenary physics professor came with him, and they were looking at it, and I was showing, and they were going, "Wow, you guys do science?" And it's like, yeah. And so, of course, everybody likes to ask questions about exercise. And so he said, I noticed, he was a runner, and he said, I noticed that when I take a few deep breaths before I start exercising, I thought it would just help for a little bit, but I take a few deep breaths and I feel good for a really long time, okay? And I just started thinking, okay, you reset the starting point, and then the exercise ventilatory response regulates whatever that new starting point is, okay? I thought, that's a testable hypothesis, okay? I can do student research with that. So with the goats, we increase the starting point, okay? If you're trying to lower the starting point, well, you can't really ask a goat to hyperventilate, okay? But you can ask a human, okay? So the hypothesis then, instead of elevating the starting point, we're going to try lowering the starting point. So humans are unusual in that we maintain what's called isocapnia, the same CO2 level from rest to exercise. By the way, when we talk about exercise, we're talking about mild to moderate exercise. We're just talking about enough to increase breathing during exercise. So, walking 3 miles an hour, okay? So anyway, the regulation relative to the resting level in humans is the same, isocapnia. Most animals, most mammals actually decrease it. Their exercise ventilatory is pre-programmed. So our hypothesis is, well, if we lower the starting point, okay, we should see a lower exercise CO2. Now, arterial blood samples are frowned on by most human subjects committees, okay? And also you need a blood gas analyzer. Those things are messy and expensive, okay? But what we can do is you can measure CO2 at the end of a breath, okay? That reflects what's in the lung, okay? So, End Tidal Co2. So the way that's done, this is the breathing mask. Here's a little End Tidal CO2 port, and it's connected to a separate little CO2 analyzer, okay? And what it does, it detects the peak, okay? What the highest CO2 level coming out of the mask is, and records that, okay? So you can indirectly get at arterial CO2, and in that case, it's like get an idea of what's going on in the lung, which is controlling what's going on in the blood, okay? Okay, so alveolar CO2, or the CO2 in your lung, is what determines arterial CO2, or PCO2, CO2 pressure. So we started out, and I started this research at Hendrix College, where I was before, and so we did the experiments. There was a lot of troubleshooting to be done, and so what this is showing is End Tidal CO2 at rest and then after hyperventilation, okay? So hyperventilation, which in that case consisted of 5 deep rapid breaths. It's hard to sort of standardize how much hyperventilation goes on, but we were looking for a significant decrease, and as soon as they did that, they started exercising. Okay, so the change in End Tidal CO2, CO2 when you exercise, you're taking bigger breaths, so the peaks are higher, the lows are lower. We only looked at the peaks, and so End Tidal CO2 is going to increase during exercise, but if they hyperventilate, we should see less of an increase, okay? Indirectly measuring with End Tidal CO2. So this is just with no hyperventilation. So this is oxygen consumption. VO2 is just your rate of oxygen consumption. When you exercise, your rate of oxygen consumption increases, and so did End Tidal CO2. Well, when they're hyperventilated, you really can't see a lot of difference here. But if you look at the slopes, if you do a bar graph of the steepness of the slopes, well, okay. Yeah, this is a bar graph of all the white slopes. And this is a bar graph of all the red slopes, okay? So you can see a little bit of a trend, okay, that's consistent with the hypothesis, but there's nothing there, okay? Now, we learned a lot, okay? So we had to figure out the protocol. We'd never played with this equipment before. And so we just thought, I want to try this again, okay, with what we've learned, okay? So we tried it again. Here's the results from the first summer, and here's the results from the second summer. And again, you know, if you do a bar graph of all the white slopes compared to the red slopes, you see a difference, but it's not significant, okay? So actually I was drinking beer at a bar, talking to a guy who worked at Little Rock in a cardiac lab. He suggested a minute-by-minute analysis, looking to see what happens in each of the 5 minutes, okay? So we did that, and okay, so no significant effect on end tidal CO2, but doing a minute-by-minute analysis. Okay, this is the first minute right after they hyperventilated when they first started exercising, and darn it, it looks like there's something there, okay? But it's just tough to get at. So this is the first minute and the 5th minute, okay? This is every minute. So we can see our resting, our end tidal CO2s are pretty much the same. And then this looks like some kind of effect. Is there any way to get at that experimentally? Well, it's kind of like if this is what you see during exercise, if you have somebody hyperventilate but then not exercise, okay, and compare that to somebody who just breathed for 5 minutes, okay, you shouldn't see this difference. This difference has to be due to exercise, okay? You should see a complete overlap between these points, okay, if you're just doing it at rest and there's no exercise, okay? So we did a more elaborate protocol. We had to add resting measurements, ventilatory measurements, to these exercise measurements. So before we could get subjects to volunteer, but then we had to start offering $10 Purple Cow gift cards, which is an ice cream hamburger shop close to campus, and our new protocol for randomized conditions. So these were done in no particular order, just to prevent that effect. 1, regular exercise, that's called eucapnia, so no hyperventilation prior, okay? And then hypocapnic exercise, okay? So we just made sure we jumbled these conditions up for every subject, okay? So that was hyperventilation prior to exercise, and then regular rest or eucapnic rest, where they just sat there and breathed for 10 minutes, okay? Because before exercise, we got 5 minutes of baseline data. So we just did the same thing for the resting ventilation, the resting values. And then a hypocapnic trial where after 5 minutes they took, in this case we were doing 7 deep rapid breaths. So that way we can compare the differences in the resting levels to the differences in the exercise levels, okay? And so again, we spent all summer recruiting subjects and getting the number, doing the, trying to get good data. And so, wow, that was a lot of work. The results, I was thinking about putting WTF at the top there, but because this was really disappointing. Our baselines were just all over the place, okay? We were just really getting unreliable results. So this is just normocapnic rest, these squares here, okay? Or yeah, the red squares are normocapnic rest, and this is where they hyperventilated. But thing is, we got some artificial significance just because the baseline started at different places, okay? With exercise, it's kind of like, yeah, it's like, what do you do about this, okay? So baseline end tidal CO2 was all over the place, and as it turns out, well, I found this out when I got this. They actually sold my end tidal CO2 module to analyzer to somebody else. So they sent me one later on with instructions on how to hook it up. They told me everything except that there needs to be a filter at the intake there, okay? That filter can make a big difference, okay? And apparently it did. So we got all of our data, and the baselines were all over the place, and it's like, gee, that's nice to know now after we've done all of our experiments. Okay? But anyway, is there anything that can be done? Well, the way I kind of, may do is to look at the percent change. Okay? So basically, you just set all of these baselines at 0. And you look at the percent change from there. Okay? So that way you can kind of standardize everything. So when we did that, we could put it all on one graph, and here we are. Okay? So everything's normalized to 0. We looked at percent change, and this is hyperventilation, okay, for both rest and exercise. So that significantly lowered arterial CO2. And then we compared these differences to these differences. Okay? So if these differences were because of exercise, you should see CO2 coming, end tidal CO2 coming back to normal much more quickly, okay, at rest, because there's not that feed-forward exercise ventilatory stimulus. Okay, and so once we got out here, well, okay, we were doing all the statistical gymnastics we could to see if there was significance. Okay, finally, on a one-tailed t-test, okay, we saw a difference between what happened during exercise and what happened at rest. And it's kind of like, who cares? I'll take it. Okay. I mentioned that one of the tricks is to get consistent hyperventilation to get the same decrease in end tidal CO2 at rest, before rest when they're sitting down, or at rest, and before exercise when they're standing up. So we managed to do that. One of the questions you always get with these experiments is, are there gender differences? Okay. And it's like, okay, somebody's going to ask us that, so we better get an equal number of males and females so we can compare them. Let's compare them. Whoa! Holy crap. Okay. Yeah, this is females, okay? Big difference, okay? So we got significance, and this is only an N of 10, okay? So we can see, a marked difference with the females, whereas males, okay, not much happened, okay? Not much evidence of, feed-forward regulation to regulate relative to that resting level. And when I thought about it, all the goats we used were females. Okay. And so, yeah, all of these differences were because of the females. Okay? And so I was at the ACSM, and a friend came up to my poster, and he asked about gender differences, and I pulled these graphs out. I had them in my briefcase. We didn't have them on the poster. He goes, you mean to tell me that all of this is because of what you saw with the females, and you didn't put that on your poster? And I'm like, no. Okay. But anyway, so that's where it stands now. And so future directions, my colleague, I presented this when I was interviewing here. She goes, this could have to do, and people said, well, what do you think caused the gender, the difference in females and males? And I said, that would be explained by what's called a gender difference. What all goes into that? I have no idea. There's just way too much. But my colleague said it might have to do with, the follicular phase, something like that. That might have and that she's the expert on that and is, studying that currently. And so it is very nice to have that input, that feedback, those ideas on, wow, what might account for these gender differences between males and females, because it's pretty clear. But for now, future directions include the filter on the CO2 analyzer. It's much more satisfying to have absolute numbers than to look at percent change. And just there's more data analysis that can be done, looking at more of the details of what happened to breathing at different points, ventilation, tidal volume, frequency. You can look at ventilatory drive, which is the VTTI, how long it takes to inhale a particular volume, your inspiratory flow rate, your timing component, time of inspiration relative to the time of the respiratory cycle, which can get at, okay, is there something happening to the pattern generator compared to the rhythm generator? Is there something going on there? So there's a lot more analysis can be done. And they explore gender differences and Shannon, help here, okay? Because I don't know what I'm doing. Anyway, but that's worth pursuing, okay? So that's where things stand now, and just in terms of acknowledgments, credit, okay. Gordon Mitchell was my postdoc boss at Wisconsin, and Brad Hodgman was our lab tech. Bruce Jens and Jill Josephs were both vets. Well, he was a vet student. She was on the barn crew, but eventually went to vet school. And then Andrew Kepper was actually a high school student who worked in our lab one summer. It was a lot of help with the systemic 5-HT, by the way, 5-HT is 5-hydroxytryptamine. It just means serotonin. I'm sorry for not explaining that, okay? But he was part of those experiments and was just ended up being wonderful. And as far as the research I did at Hendrix, well, the first summer where we were trying to get everything figured out, doing a ton of troubleshooting, Maitri Shah and Elizabeth Krug, she's in med school now. She was, Elizabeth was, she was Division III national champion in the heptathlon, so she was always willing to jump on the treadmill and run for as long as we needed to try and figure stuff out. And it's like, let's try this. So that was nice. When we decided to try it again, okay, these three students worked with me one summer. And then the last, so I think she's in med school now, Van Fung. And last summer where we did the resting measurements in addition to the exercise measurements. This is Ella Konkavich-Nassar. She's actually in PT school at Methodist now. And this Connor, I need to find out what happened to him. But other than that, anybody have any questions? So, yes.
I was just wondering, does age have a bearing on these in terms of their variables?
It could. It could. That's another potential avenue of research. So, yeah, when this first started, we used goats because they're a good representative mammal, okay? You can take repeated blood samples. They have a breathing pattern similar to humans, okay? Dogs tend to pant. Plus, a lot of people like dogs, including us. Plus, they're big enough so you can take repeated blood samples to get arterial blood gases. We didn't really, we used the culls from the dairy herds up in Wisconsin, okay? I'm not sure how much age difference there was in the goats or anything like that, but there certainly might be potential there, yeah. But yeah, I think aging would have to do more with what I was talking about earlier when something happens and it's harder to breathe during exercise and you need that boost from serotonin. At least that's what I'm experiencing. Okay. Okay. Yeah.
This may be out in the field. Do you think there could be any validity to the idea that getting good respiratory data is so hard because it's our involuntary voluntary system? For instance, your heart rate goes up, but you don't think, okay, I might want my heart to contract stronger at the same time. It's pretty much an involuntary system. Whereas the respiratory system, we don't think about taking every breath, but we have voluntary control. It's skeletal muscles. So you're bringing in higher brain centers at the same time and changing rate and depth of breathing, so you got two variables going at the same time. Could that play a part in why you got your numbers going all over the place?
I had to do more with the fact that End Tidal CO2 is, it is difficult to get an accurate measurement there. And, you know, it's like if you're really talking, it's like a difference of 2 millimeters of mercury, that's not a very big difference. And it's real tough to get at with End Tidal CO2. So there's going to be a little bit, you know, of slop using End Tidal CO2. And, you know, it was even worse just not having the filter on the End Tidal CO2. And you can get at that, you know, just by having a larger sample size. You know, if you get that small difference with a large enough end, then, you know, you can show a significant difference. But for what we were able to do in one summer, you know, we were able to do 20 subjects. And more subjects, we might have seen a difference. Well, plus we got decent measurements. You know, we might be able to see a difference. But and at this point, I forget whether I'm answering your question or not. Okay. Okay. So, yeah, John.
When you first mentioned that the hyperventilation before exercise might kind of reset things in a positive way, I wondered if yawning before exercise Because I know when I go to the gym and work out, when I'm walking into the gym, I often yawn 3 times. I get ready to go to sleep pretty quick.
Yeah, right.
So I don't know much about the physiology of yawning, but I just wonder if y'all ever thought about that or read about it anywhere, because it's obviously a way to hyperventilate in the short term.
Yeah, of course, I always thought of yawns as you're tired and your breathing is insufficient to regulate CO2, so your body kind of goes to reestablish the normal level as opposed to hyperventilating.
Right. I
But
just wonder, I know sometimes people will yawn when they're nervous, so I'm wondering if it's like a way to feed forward for some sort of stressful situation that's coming up.
yeah, I guess I'm more inclined, that does not mean it's right, but I'm more inclined to think of the fact that they're nervous, they're focused, and their breathing is suppressed. So arterial CO2 kind of builds up, and you kind of get that urge to take a deep breath to reestablish. But like I say, that's just my feeling on the matter. So objective feeling assessment. Okay. Yeah. Okay. Not exactly scientific. Okay. Okay. All right. Okay. Yeah.
Altitude affects that?
Altitude. It's. Let's see. Yeah, altitude. That's an example of where you actually do have. I mean, you get small changes in CO2. You get a couple of millimeters arterial CO2 change. You see a big effect on breathing. Oxygen, it has to get down you have to drop it by like 40 millimeters of mercury, from about 100 to about 60. Then you start to an increase in breathing because of oxygen. So you have to get pretty hypoxic. So yeah, if you're at altitude, depending on how high you are at altitude, at the Olympic Training Center, I remember being on the treadmill, and you could feel the effects of altitude. The interaction between oxygen, we actually did everything to make sure that oxygen was not a factor. We gave them a hyperoxic mixture. So when they were breathing had the dead space, they were rebreathing CO2, but they had plenty of oxygen there. So we actually tried to eliminate that. It's possible. It seems like it would be some kind of interactive effect with oxygen playing more of a role as you went to higher altitude. I really can't give much better of an answer than that. Okay. Yeah.
Did you do anything involving anything which involves like checking body fat percentage in regards to this experiment?
Yeah, no, I think what that would have affected was the calculation for the rate of oxygen consumption. So we did look at VO2. So VO2, the units for that are milliliters of oxygen per kilogram of body weight per minute. And so, yeah, basically if you have a higher percent body fat, that's going to lower your rate of oxygen consumption just because fat's not metabolically active. But most of our, most of our subject population was fairly homogeneous. Okay, like 20 years old, plus minus, 1 or 2 years, and then, their fitness levels appeared to be somewhat consistent.
I was thinking because of the difference between males and females that it could have had a role, possibly.
Yeah, females do have a different percent body fat, typically. Let's see. I think we, again, would probably see that with a calculation of oxygen consumption with VO2. I don't recall that. Yeah, that's another place where, we have that data. We could go back and look at that to see if there were differences in, significant differences in metabolic rate for an equal workload, because everybody was walking at the same speed. Interesting question. Great. Okay. And I'm not going to waste your time if you don't want to ask me questions. Okay. Thank you.